Today we are going to start with the topic somatic embryogenesis. Somatic embryogenesis, generally plant embryogenesis. What is embryogenesis? It is development of an embryo from plant cells or a zygotic cell. What is somatic embryogenesis? Somatic embryogenesis is again I have to say, it is the development of embryo from somatic cells. A gist of what is written in the slide. Okay, these are the pictures. These pictures are indicating, the first picture is callus. Through callus it is propagation of plantlet, that is in the second this picture, and overall picture of somatic embryogenesis, and a plantlet is shown in these pictures. Brief historical background. There are two scientists who has worked initially on this somatic embryogenesis, was Stewart and Rennert. Stewart was the one who has worked on embryogenesis in suspension culture, and Rennert person who has worked on somatic embryogenesis on callus culture of carrot. The next slide tells you about the types of embryos. What is the types of embryos? Zygotic and non-zygotic are the two different types of embryos. Zygotic is about all the reproductive parts of the plant, that is fertilized eggs, and here non-zygotic embryos. Today we need to talk about somatic embryos, so we need to understand whether it is a zygotic one or non-zygotic one. So somatic embryos are the embryos which are derived from somatic cells, okay? And there are two other non-zygotic embryos. One is parthenogenetic embryos and androgenic embryos. So what is parthenogenetic? Unfertilized eggs. Can you give one example of that? Seedless? Grapes. Very good. Androgenic embryos. Androgenic embryos is formed from male gametophytes. So what is the example for this? If parthenogenetic embryos is for seedless, androgenic embryos is GM crops. So that is one, and also it appears in vivo conditions. Okay, so that is the types of embryos, zygotic and non-zygotic. You need to understand these two embryos, zygotic and non-zygotic, before entering into the actual process of somatic embryogenesis. Is that fine? So next subheading of this topic includes the phases. What are those phases? Inductive and expressive phase. In inductive phase, we need to have the initial stages that we need to understand in any of the plant tissue culture process. What is this inductive phase? Inductive phase is the initial stage as it is explained in the slide, and it is the induction of the development of an explant. What is an explant? One at a time. Anybody can say what is an explant? Sai Kiran? Used for inoculation. Very good. Please sit down. So using an explant in vitro condition to inoculate, and that is developing into a globular structure, okay? Globular structure, that is inductive phase. The development I have shown you a picture here. One is ovular shape, that is nothing but globular shape here, and the next is heart shape, next is torpedo, next is the cotyledonary. This is the overall picture of somatic embryogenesis. In this phase, the inductive phase, what I am talking about is the development of an explant into a globular structure is the first stage, and that is the only phase where induction takes place. Next phase is expressive phase, in which we will be studying about development and maturation. So this is about globular stage. I was telling about inductive phase, where this is a callus, or this will be, globular stage will be like a callus which is globular in shape. So the next stage is expressive stage. The arrow mark, the flow chart that is shown, started with inductive phase here and then continued with expressive phase. So development phase of expressive phase includes two structures. One is heart shape, another is torpedo shape. Now you can see in the picture here, this is looking like heart shape. Can you see this? Little bit editing has been done in that. So it looks like heart shape and green in color, right? Green in color. So that includes the somatic cells and also indication of the development of an explant, okay, towards somatic embryogenesis. So this is heart shape, and then the next shape is torpedo shape. Can you see what is torpedo? Torpedo is missile or submarine, general meaning. Yes. So isn't it looking like a submarine with two cotyledonary shaped structures which are going to develop into leaf later on. So this is a torpedo shaped structure which is going to initiate and then develop into a shoot. And then maturation stage of expressive stage phase includes two different structures again, that is cotyledonary and germination. So this is the cotyledonary stage. Okay, this further develops into a plantlet, and then finally the embryo becomes cylindrical and then leads to germination. This is the complete process. Did you understand this? This is the flow chart of entire two phases. One is inductive phase, another is expressive phase. Got the phases of a somatic embryogenesis? Yes. What is the next information that you need to understand about somatic embryogenesis is about factors affecting somatic embryogenesis. As you have been studying in plant tissue culture, the factors that do affect for other cultures like protoplast culture, meristem culture, anther culture, endosperm culture, the general factors are pH, temperature, humidity, light. Apart from that, nutrition is the one which gets modified dependent on the explants that we are using. So what is the specificities of factors? So pH is 5.6 to 5.8, temperature 26 to 28. Yes, humidity 40 to 80%. Then in light, there are two different criteria. One is intensity, another is duration. Intensity is? Yes, it is expressed in units, that is 5,000 to 8,000 lux. And duration is? 16 hours light. Yes. And as I have been telling you, the modifications of the nutrient composition is dependent totally on the explants from which it has been derived, with species. Okay? Two modifications that are needed is explant and the nutrition composition. Further, for regeneration of somatic embryogenesis, there are two types of somatic embryogenesis. One is direct embryogenesis, another is indirect somatic embryogenesis. Usually, if we have to define this embryogenesis or any other part of plant tissue culture, there are two methods that are followed in vitro condition. One is direct, another is indirect. Direct is a process of plant tissue culture in which the development of somatic embryos takes place without intervening of callus. That is direct somatic embryogenesis. That is, we can see in pictures, you see here, there is explant in this plate, Petri plate. It has been inoculated in the nutrient media, and that is showing into a plantlet here, like germination in case of seeds if you sow, and it shows germination, that is the beginning. And here in vitro condition, you can see there is a plantlet from the explant, and then there is direct plantlets that are growing in the picture C, and in picture D you can see the development of plantlets in initiation of roots here. Okay, that is direct somatic embryogenesis. Next is indirect somatic embryogenesis. This is the process in which you develop an explant, surface sterilize, inoculate it, and that explant develops into a plantlet with interference or intervening callus. That is, explant gets developed into an plantlet with via, you can say, via the interference of callus. So, to explain in the picture, this is the explant. After surface sterilization, you have inoculated in the nutrient media, and then it is developing, propagating after few days. Then it is developing into callus. Callus is also further developing into a plantlet. You can see here, yeah, this is the callus here, and the plantlet is growing from callus. And this is the, again, it has been subcultured. This plantlet has been removed, and then it has been subcultured into a fresh nutrient media, and this further rooting has been takes place. What are the stages of micropropagation? All the stages you can see here, right? Starting with selection of the explant, establishment of the culture, yeah, multiplication here, right, and then rooting here. The plantlet which is observed in D, picture D, has been inoculated in a media where the rooting, the plant growth regulators has been given into a nutrient composition, specifically auxins. What are those auxins? Can you name few auxins? NAA, IBA. Okay, these are the auxins, and these plantlets have been transferred to a media which contains these auxins, which auxins will facilitate specifically rooting. Okay, and then it has been transferred to a pots which contains, what is the hardening media contains? Coco pit, charcoal, and sand or sterile soil. You should mention sterile soil. In plant tissue culture, sterility, not sterility, sterile conditions. Sterile condition is very important, though it has moved from laboratory to polyhouse conditions. Once from polyhouse it reaches the field, then there is no problem. But when the plant from plant tissue culture bottles moves from laboratory to polyhouse, then also sterile condition is very important. In hardening case, where these pots you are seeing in picture F, you can see there are pots containing sterile soil, coco pit, and charcoal. Okay, this is hardening, and this complete picture of indirect somatic embryogenesis indicates that explant develops into plantlet via the interference of callus. Okay. Next is the applications of somatic embryogenesis. What are the applications that you have so far studied with other cultures? Is usually disease resistant plants, right? Disease resistant plant production is one application, and it also applies with somatic embryogenesis. Okay, and further is there is production of homogeneous plants. There is in some cases what happens, the plants will not have reproductive paths. In that case, you can isolate the protoplasts, and also you can subject that into cultures for the development of embryo through protoplast. That is also one possibility. Protoplast culture, through protoplast culture you can develop somatic embryos, and saving the rare and about to get extinct plants species is possible from somatic embryogenesis, and this is of high rate multiplication is possible with this process. And further we will also talk about the another topic of plant tissue culture, that is protoplast culture. What are protoplasts? It is a basic fundamental information that all undergraduates should know. What is a protoplast? Durga Malleshwari. Plasma membrane and other cellular organelles. Please sit down. So that is protoplast, and we are talking about protoplast culture today. So in protoplast culture, we have to study about protoplast isolation, protoplast fusion, identification and selection of protoplasts that has got fused, and then culturing, also regeneration of protoplasts. So this is the overall picture of protoplast isolation. This picture, what is indicated here, this is one explant in which the epidermis will be there. The cells are arranged like this, and there is a microscopic observation that is going on, and this is knife is shown. Here in protoplast culture or protoplast isolation, knife is shown there. So knife is nothing but your mechanical and enzymatic method. Okay? Mechanical method usually there is agitation that is applied on an explant. What kind of agitation? Agitation can be any kind of force or any kind of pressure that is applied on an explant. Okay? Here they have shown knife. Usually knife is not a actual method to cut a cell, because that is cell is a microscopic, and we need to subject an explant either mechanical method or enzymatic method for this cell wall to be removed, and then this cell has to be isolated like this. So in detail, to study about protoplast, let us understand about the historical background, who has contributed what in protoplast culture. So this is, there is one scientist by name Hanstein put forward the term protoplast. Okay. So later on there were two scientists, Klecker and Cocking, who isolated the protoplast by mechanical method and enzymatic method respectively. These three scientists are the main contributors, and if you remember these scientists for your syllabus point of view, that is fine. And next is, as I was telling you, the protoplast isolation, two methods are carried out, that is, one is mechanical method, another enzymatic method. Now mechanical method, this mechanical method, I said there is agitation that has to be applied on an explant. So what is agitation? What is agitation? Force or any kind of pressure that is applied on an explant. Take an explant, maybe it is a leaf or stem, it is subjected to grinding. Usually in laboratory level, what is grinding? How do we do it? We do grinding of an explant by using mortar and pestle. Grind the solution, and this solution is transferred to another beaker or a test tube through filtration. Filtration is done by either a nylon cloth or any filter papers. So once the grinding is done, filtration is done, and then subject the solution into centrifugation. Centrifugation is done with a specific RPM, maybe around 2000 to 8000 RPM for a period of 15 minutes to 20 minutes. And once the centrifugation is completed, the protoplast is collected in the supernatant or pellet. We are subjecting this mixture in centrifugation, that is specific centrifugation. What is that centrifugation is called? Density gradient centrifugation. Density gradient centrifugation is the centrifugation method that we are using in mechanical method, and then the pellet will have the protoplasts that are isolated, not the supernatant. Supernatant is collected only when we are isolating some pigments and not the cells. Pigments will get collected in supernatant, and the cells, if we are isolating, it gets collected in pellet, and it is dependent on density gradient centrifugation. The next method is enzymatic method. Here, as we all know, the explant, first of all, it should be subjected to surface sterilization. Surface sterilization is done for any explant to remove dust basically, and then it is taken into laboratory for further surface sterilization to remove microorganisms, as I said. But then we are going to use some surface sterilization chemicals like mercury chloride and sodium hypochlorite. Mercury chloride is used to remove bacteria, and sodium hypochlorite is used to remove fungus. So that is the main purpose of surface sterilization, which is done in the initial stages. And next, enzymatic method we will study for protoplast isolation. In this method of enzymes, we need to study about two different steps. One is two-step, another is one-step method. Two-step method is also called sequential method, and one-step is simultaneous method. In two-step method of enzymes, we have to study about the method where explant is treated with two different enzymes, one after the other. The explant is treated with pectinase to degrade pectin, and the next step is the explant is treated with cellulase. And over this two-step method separately it will be followed, and that is why it is a sequential method. Pectinase is given a treatment to the explant overnight. It may be ranging from 12 hours to 20 hours, and then the explant is transferred from the mixture of pectinase to cellulase, and this is from minutes to hours. Cellulase treatment is given to isolate protoplasts further. This is two-step method, which is also called sequential method. In one-step method, which is simultaneous method, the explant is treated with a mixture of enzymes. This mixture of enzymes consists of macerozyme and cellulase. What is macerozyme is also called pectinase. This is otherwise called pectinase. In one-step method, which is simultaneous method, the explant is treated with a mixture of enzymes, two enzymes. One is macerozyme and cellulase, and you can extract protoplasts. So the next slide is telling you about the techniques of protoplast culture. You have isolated the protoplasts and you need to culture those single protoplasts. Why do we need to culture the protoplasts? Why do we need to culture the protoplast is a question that arises usually when we talk about the technique of protoplast culture. So we have isolated single protoplasts, and now what we need to use that protoplast for? We need to use that for further propagation in plant tissue culture, right? So what is this further propagation? Protoplasts are microscopic in nature, in the sense after we isolate it, and they are not seen to the naked eyes. They are only single protoplast can be observed under microscope. Okay, we can only see the pellet, that is group of protoplasts, not single protoplasts. They are observed under microscope. Now, the techniques of protoplast culture is a technique. There are many techniques that we need to follow under protoplast culture. We are going to study about few main cultures here. One is liquid media and another is solid media. One is feeder layer technique, another is co-culture, and one last is microdrop culture. Okay? So, let us go one after the other techniques to culture protoplasts. These are microscopic in nature, and they have to be handled very carefully. And while handling, these should be transferred to cultures for further propagation. So that one method is they are cultured in liquid media, that is otherwise called suspension culture. Yes, a liquid media in plant tissue culture is otherwise called suspension culture. So we inoculate this protoplast in liquid media and get, as we were talking in mechanical method, we centrifuge it and then take the pellet, and then we get a lump sum amount of protoplast, and then we can directly culture it on the protoplast on nutrient media in plant tissue culture bottles. And the second method is on solid media. We isolate it, centrifuge it, get the deposit of protoplast and culture it on solid media. So that will give the propagation of protoplast. And further, the third method is feeder layer technique. Feeder layer technique is the one you must have heard about nurse cells. Nurse cells which absorbs nutrition from the source and supply it to the offspring or the explants. Here in plant tissue culture or protoplast culture, we have this technique, feeder layer technique. You can observe here there is a layer of nutrient medium, and in between, that is on the top of the nutrient media, there is a feeder layer. This yellow color mark you can see on the top of nutrient media. This is a layer. This layer is nothing but nitrocellulose membrane. This membrane, on the top of this membrane, we have to place the protoplast. The green colored structures here, globular structures, are protoplasts, and it is labeled also, are placed on nitrocellulose membrane, and here the nurse cell role is played by nitrocellulose membrane. This layer absorbs nutrition from the media and supplies it to the protoplasts. That is how they get propagated further, and once after a few days, maybe around 10 to 15 days, they are further transferred to plant tissue culture bottles with specific composition. This is about the feeder layer technique. The next co-culture method, this method is also a method in which it has nutrients in it, a layer of nutrients, and then placed protoplasts in this. This picture represents the protoplast, that is two different protoplasts. One is slow growing, another is fast growing. That why slow growing and fast growing protoplasts are placed alternatively. There are alternative protoplasts that are placed. One is slow growing, another is fast growing protoplast. That is, maybe we do not know unless we culture it whether they are fast growing or slow growing. It is only that two different species, that is two different protoplasts isolated from two different species have to be placed alternatively. In that case, there is no competition that takes place for absorption of nutrients. Okay? What is it? There is slow growing one and fast growing, here they have represented. If it is placed like that, it is only that there is no competition for these protoplasts for the absorption of nutrients. Got it? Yes? The next technique is about microdrop culture. Microdrop culture is a tank in which the tank is divided into two. One is contains full of sterile distilled water, another is subdivided into micro wells. These are micro wells here. You can see the divided, it looks like cylindrical structures. These are micro wells, and this is where sterile distilled water is placed. What is, why sterile distilled water is placed here? To maintain humidity. And these are the micro wells. Below this, there is a place that is given. This place is filled with nutrient media. Nutrient media, and each of these micro wells are fitted with, are having opening in the bottom, and they are fitted with a mesh. And these meshes have such pore size where they are in which we can place microscopic protoplasts. Microscopic protoplasts, you can understand, there is its structure may be around nanometers. Each of these wells here, okay, that these wells will contain only single protoplast. That means you can place only single protoplast in each of these micro wells, and there is no competition between each protoplast here. It has been completely isolated. One chamber is given for single protoplast, and there is absorption from nutrient, it takes place, and then they propagate. After few days, like I was saying, 10 to 15 days, they will be transferred to plant tissue culture media, that is bottles which contains nutrient media. So these are the techniques for protoplast culture. After having known about the protoplast isolation and protoplast culture, it is that why we are isolating protoplasts and why we are culturing protoplasts is a question. Why is that so? So that is answered in this slide, that the applications of protoplast culture is shown. What is the protoplast? You have all have known, and you also have got to know how it has to be isolated, and further how isolated single protoplasts will be cultured and then used for micropropagation of various other parts of the plants, right? So here you can see there is production of cybrids. Application of protoplast culture will lead to the production of cybrids. What is a cybrid? Cybrid is a fusion of two cells with nucleus of both or single parent. Cytoplasm of both the parents get fused, and that fusion will lead to the formation of cybrids. That is cybrids. And why this production of cybrid is possible from protoplast culture, and why it is so? A desired character is there in two different parents. For instance, parent number 1 disease resistant, parent number 2 is high yielding. So you need those cytoplasmic characters or traits to reproduce in a single offspring. So in that case, we need to fuse these two cytoplasms. So in that case, we need to do this cytoplasm culture and produce hybrids. Then further, maybe in the first slide I have written somatic hybridization. So, protoplast fusion is otherwise called somatic hybridization. Why somatic hybridization is necessary? To overcome a problem which is sexually incompatible between two parents. So, the somatic hybridization or protoplast fusion is necessary and very much essential. Okay? When I was talking about this also, when reproductive organs are not compatible, then this protoplast culture or somatic embryogenesis or somatic hybridization is necessary. The next is DNA transformation. DNA transformation is possible through protoplast culture if there is no possible facilities that are available in the laboratory. And then is single cell production. What is a single cell culture? Can you see here, there is a clumps of cells, okay? And you are subjecting it to, maybe you can consider here as a mechanical method, you are subjecting the explant for mechanical method. And then you are isolating protoplast, is also called single cell culture. So, that is possible through protoplast culture. And finally, the resistant plant production is another important application with these three processes. What are those three processes? That are maybe two processes we are talking about today in plant tissue culture techniques. One is somatic embryogenesis, another is protoplast culture. So, disease resistant plant production is the main objective for any of the plant tissue culture techniques. Is that fine? So, understood about somatic embryogenesis and protoplast culture today? Yes, madam. Thank you.